Basic medical sciences, welcome back. So this is the third and last video on microbiology Krok-1 series. Right, so let's continue. After sensitization, a test animal received subcutaneously a dose of antigen. At the site of injection, a fibrinous inflammation developed with alteration of vessel walls, basal substance, and fibrous structures of connective tissue. The inflammation took form of mucoid and fibrinoid degeneration, fibrinoid necrosis. What type of immune response occurred in the test animal? Right. So, firstly, after sensitization, the antigen, another antigen, was introduced into an animal. Right. So, the kind of this is a re-exposure to an antigen is type 1 hypersensitivity reaction, also known as immediate hypersensitivity. Brain autopsy revealed an edema, hyperemia, and small hemorrhages of medulla oblongata. Microscopically, chromatolysis, hydropia, and nerve cell necrosis are observed. With the cytoplasm of hippocampal nerve, there are eosinophilic structures, Negri bodies, detected. What diagnosis correspond with the described morphological signs? Right. So Negri bodies is a buzzword for what? Rabies, right? So it's rabies virus, right? So here we are talking about an autopsy, right? So in most cases, you should know that Negri bodies are found in hippocampus and in the pyramidal cells of the cerebellum, right? Okay, so this is a random slide from the rabies video, right? So on the question, it was about an autopsy, but what about in a living person, right? So for diagnosis, firstly, we'll talk about history of exposure to a potentially rabid animal. Laboratory diagnosis includes in serum, non-immunized patients will find rabies antibodies, and immunized patients, there will be rising serum antibodies over a few days. In the cerebrospinal fluid, we'll find the characteristics of encephalitis. Skin biopsy from the back of the neck for polymerase chain reaction, right, is reverse transcriptase polymerase chain reaction and immunofluorescence testing. And we can also do it in saliva for viral RNA and viral culture, right? So you know what you have to do. After the end of this video, you can check in the description box. There is a link to RNA viruses playlist, so you can watch those videos in order. That will be very helpful. Pathologic material, mucosal secretion from the nasal passages obtained from a patient provisionally diagnosed with influenza was delivered to the virological laboratory. What quick test allows detecting specific viral antigen in the investigated animal, right? So quick test, if you find this password, quick test is immunofluorescence, right? So quick test, immunofluorescence, right? So you just have to check here, and you only have direct and indirect immunofluorescence. The rest, there is no immunofluorescence here. ELISA, hemagglutination, blah blah blah, right? So quick test, immunofluorescence. A patient with pneumonia has body temperature of 39.2 degrees Celsius. What cells are the main producers of endogenous pyrogen that had caused such a rise in temperature? Right. So you should know that in case of inflammation, there are specific cells which produce interleukins like interleukin 1, interleukin 6, right? Interleukin 1 will go to the hypothalamus and it will cause temperature resetting, that's causing fever. It will increase the body temperature to make sure that the temperature of the body is like not comfortable for the uh, microorganism causing infection, right? So it's like a protective mechanism, right? You know what else is produced by monocytes is interleukin 6. Interleukin 6 will go to the liver and stimulate the synthesis of acute phase reactive proteins like C-reactive peptide. And another one is tumor necrosis factor alpha, right? This tumor necrosis factor alpha will go to the bone marrow and stimulate the synthesis of white blood cells, that's leukocytosis, right? So don't be confused, right? Monocytes are like precursors of macrophages, right? So they can just change monocytes and put macrophages, right? So it will be the correct answer. Okay. Gram-positive spore-forming bacilli were extracted in anoxic environment from the patient's wound contaminated with soil. Cultivation of a blood glucose agar, not cultivation on a blood glucose agar resulted in growth of the colonies surrounded with hemolysis zones. What agent was extracted from the wound? Right. So, motorcycle accidents, gunshots, those wounds are specific for Clostridium perfringens. Right. So this is Clostridium perfringens. And the other way, right? So you see, spore forming microbes, you just need to look for there are two kinds of bacteria which produce spores, right? Bacilli and Clostridia species. So you have two, and then you find a way to eliminate botulism, because the one which causes this hemolysis in most cases is Clostridium perfringens, and is also found in contaminated soil. Clostridium botulinum, canned foods, right? So don't confuse it there, right? So a random slide from Clostridium perfringens videos, right? So it's about virulence factors. So you will find three main virulence factors. The first one is alpha toxin, or lecithinase, or phospholipase C, right? So this one is the one responsible for hemolysis. It's lethal and it causes dermonecrotic properties, right? And then there are proteolytic enzymes and the polysaccharide capsule for antiphagocytosis, right? And again, this bacteria produces a gas, right? So you can actually do percussion on top of the wound. There will be like a tympanic sound, right? And this bacteria is very dangerous. You may need to do surgery, like to eliminate the necrotic tissue, right? Okay, so watch that video, right? It's very helpful. Typical manifestations of food poisoning caused by Clostridium botulinum are double vision, abnormal functioning of swallowing and breathing. These symptoms develop as a result of what? Okay, we covered this question before, right? So botulinum toxin is an exotoxin, it's a neurotoxin, right? Okay. On examination of a 6-year-old child, the doctor noticed grayish film on the child's tonsils. Microscopy of the smears stained by Nissa's method detected the Corynebacterium diphtheria. What morphologic feature was the most indicative for determining the type of agent? Right, so Clostridia, Corynebacterium diphtheria is known for what? volutin granules on the pores, right? Volutin granules. A man is suffering from diarrhea. In summer, he spent his vacation in the south at the sea coast. Bacteria with the following properties were detected in his feces, right? Gram-negative curved rods, gram-negative curved mobile monotrichous bacilli that do not produce spores or capsules. They are undemanding to nutrient medium but require alkaline reaction, right? Alkaline environment, 1% peptone water, cholera. On examination of a patient with disease onset 5 days ago, the doctor suspected tularemia, right? Tularemia caused by Francisella tularensis, and prescribed the patient tularin intramuscularly. What is the purpose of this drug administration in the patient, right? So this one is more like Mantoux test, like for tuberculin. It's diagnosis, not prevention. Tularin, right? So it's just like tuberculin. It's for diagnosis. Allergy diagnostics. Initial inoculation of water in 1% peptone water. This is cholera, right? Sometimes you don't need to read the whole question, right? And we've covered this question several times. So I'm sure now you know all the buzzwords concerning cholera. Rice water, okay. In a town, there was registered an outbreak of hepatitis, which had supposedly spread through water supply. That's fecal oral route. So, do you remember acute A and E? The hepatitis viruses transmitted by fecal oral route, and also they cause acute infections, right? So, here you have it, A and E, acute infections. Vowels are bowels. Okay. A patient was hospitalized into the infectious disease unit on the 11th day since the disease onset and provisionally diagnosed with typhoid fever. What biological materials should be collected from the patient for the analysis at this stage? Right, so just to check the time period, 11th day. At this point, the antibodies are already present in the serum, right? So we take blood serum. 3 days, because a question which is similar to this one, it will just say 3 days. So in 3 days, you usually take stools, right? Okay. A patient with clinical signs of a primary immunodeficiency has functionally disturbed mechanism of antigen presentation to the immunocompetent cells. What cells are likely to have the structural defects, right? APCs, antigen presenting cells, are macrophages and monocytes, right? Okay. Macrophages and monocytes are the APCs, antigen presenting cells, right? They present the antigen on their MHC2 molecules to the T cells, and then the T cells will stimulate the B cells to transform into plasma cells, and then plasma cells will produce antibodies. You know the drill, right? And just to remind you, macrophages and monocytes produces endogenous pyogens like interleukin-1, right, causing fever. A toxin neutralized with 0.4% formaldehyde under 37 to 40 degrees Celsius for 4 weeks is used for vaccination. This preparation was first used by Gaston Ramon for diphtheria prevention. Name this preparation, right? So Gaston Ramon is definitely diphtheria, right? Because another name is called diphtheria toxin Ramon or diphtheria toxoid, right? It's an anatoxin. It's an anatoxin. Okay, so on this slide, I found it on Kaplan. It's not in one of my videos, right? But it's a classification of vaccines, right? So you have two groups here: live attenuated vaccines and inactivated vaccines, right? So in live attenuated vaccines, we have viral and bacterial. Virals are measles, mumps, and rubella, varicella, yellow fever, nasal influenza, smallpox, and oral rotavirus, right? If you remember Rotatrix and Rotatec. Okay, and then bacterial, live attenuated, you have BCG of TB and oral typhoid, right? Thus, live attenuated vaccines. And then inactivated vaccines can either be whole or fractional, right? Whole or fractional, right? So, whole are actually what? Virus, thus polio vaccine, rabies, and hepatitis A, right? And then fraction can either be protein based or polysaccharide based, right? Protein based include these subunits, okay, subunit, that's hepatitis B, parenteral influenza, acellular pertussis. And then polysaccharide based, that's where you find diphtheria here, diphtheria and tetanus, right? This one is a toxoid. And pure will be pneumococcal and Hib. This one is Haemophilus influenza type B vaccine, meningococcal, right? And conjugate, you also find Hib again, right? Hib is what? Haemophilus influenza type B, pneumococcal and meningococcal, right? So you find this it's in either pure form or conjugate form, right? You don't need to memorize this whole thing, but if you can pick 2 or 3 things there, it will work. What diagnostic method should be used in industry to test the raw leather for presence of Bacillus anthracis? Bacillus anthracis, I told you, leather or meats in marketplaces, we use precipitation test, right? So it's called what? Ascolis thermoprecipitation test is the correct answer, right? But in most cases, they just say precipitation test, right? That's the answer. During the skill building session in microbiology, the students need to stain the prepared and fixed sputum smears obtained from tuberculosis patient, right? What staining technique should be used in this case, right? Tuberculosis is caused by Mycobacterium tuberculosis. This bacteria is actually an acid fast, right? So acid fast bacteria, we use Zell-Nielsen staining method. First year school children have received the tuberculin skin test, that's Mantoux test, at the school nurse office. The purpose of this test was to determine the child that needs to receive a BCG, right? So it's diagnostic, right? It's diagnostic, and then for prevention, we'll use BCG vaccination. We covered a question like this, but in this case, it has been twisted a little, right? Why do we do Mantoux test? To determine if the person needs BCG vaccination. Preventative vaccination against poliomyelitis is made with inactivated vaccine introduced parenterally. What immunoglobulins create the post-vaccinal immunity in this case, right? So before that, I think I have a classification of polio vaccines here, right? So there are two kinds: inactivated polio vaccine and oral polio vaccine, right? Okay, so let's start with the inactivated. This one was developed by Jonas Salk. It contains formalin-killed viruses, is introduced injected subcutaneously. It provokes the IgG antibody response that will protect against the future viremia. This one do not provide intestinal immunity or local immunity, right? That's parenteral. Okay, and then there is oral polio vaccine. This one was produced by Sabin. It's also known as just Sabin vaccine. It contains the attenuated poliovirus that has lost the ability to multiply in the central nervous system. Is taken orally and precipitated and sheds in the feces in the normal fashion, but does not cause paralytic polio. It induces both local secretory IgA antibodies in the intestines and also humoral, right? Okay. So in this question, parenteral, definitely we know that is inactivated polio vaccine. But what kind of immunity? There is no local immunity, right? So you need to to remove IgA among your answers, right? So you just remove 1, 2, 3, right? You are left with IgE and IgM, right? But long-term process is IgG antibody, right? So IgM and IgG, that's the correct answer, right? Let's look at another question which is similar to this. Maybe you understand quickly, right? In our country, routine preventative vaccinations against poliomyelitis involve using live vaccines that is or administered orally. What immunoglobulins are responsible for the development of local post-vaccinal immunity in this case? Right. So, number one, administered orally, and also the local immunity, right? So, here is your classification again, right? So, local immunity is definitely what? A secretory IgA here, secretory IgA. That's the answer. Okay, right. We have another one here. Various biological preparations can be used for poliomyelitis prevention. What drug induces the type of local intestinal mucosal immunity that lasts the longest, right? What the immunity that lasts the longest is oral polio vaccine, right? Oral polio vaccine is more popular, right? It's the one which is more popular. Oral polio vaccine. Okay. MN is a carrier of HIV that is an RNA virus. The cells of this patient synthesize viral DNA. This process is based on what? Right, so you know that HIV is an RNA virus, and then when it enters into the host cell, at first it needs to be converted to DNA, right? So from a central dogma of biology, you convert mRNA to what? To DNA, and then not like that. It's DNA, RNA, protein. DNA to RNA is transcription, and then from RNA to protein, that's translation, right? So if you are reverse, you are going from RNA back to DNA, you are reversing the process of transcription, right? So it's the process is reverse transcription, and the enzyme used there is called what? Reverse transcriptase. Okay, we've seen this slide before. I took it from HIV video, right? So it reminds you that HIV virus is two copies of positive single-stranded RNA. It is reverse transcriptase icosahedral capsid with a conical, right? Icosahedral with a conical capsid, spiked lipoprotein envelope, right? So I told you this video, guys, this HIV video, I covered this virus in great detail. You need to watch this video, right? Okay. A 40-year-old woman was diagnosed with glomerulonephritis based on her clinical symptoms and the results of urine analysis. Anamnesis tests chronic tonsillitis. Chronic tonsillitis. What microorganism is this? Right. So this again is post-streptococcal glomerulonephritis. Right. Yeah, it's one of the immune-mediated diseases caused by group A streptococcus or streptococcus pyogenes. And what else? Which other disease is caused by this same bacteria? Immune-mediated. Yes, it's rheumatic fever. Okay. So here the answer is streptococci. Streptococci. Okay. So this is our last question, right? Okay, let me read first. Sanitary assessment of a pond where the children from a recreation summer camp take their swims and detect their oval cysts like 50 to 60 microns in diameter with two nuclei visible in their cytoplasm, macronucleus and micronucleus. What protozoa do these cysts belong to, right? So macronuclei, like two nuclei is Balandidium coli. Sometimes they tell you that the protozoa is ciliated. So Balandidium coli is the only protozoa that is ciliated. Another buzzword is associated with pig's toes, Balandidium coli, right? So actually this question is under medical biology, right? It's in medical biology. You can watch that video. You can watch that video. It is everything in parasites, general biology, etc. Right? Okay, so no, the tick is on the wrong place. It's Balandidium coli, right? Okay, so you can see here on A is a trophozoite with cilia, right? And you can see the two nuclei, micro and macro nuclei. The second one here on B is a young cyst with a macronucleus, and you can see again here the mature cyst with a micronuclei and macronucleus, right? Okay, thank you so much. I hope you enjoyed this series, right? Thank you for watching, and please, after watching these videos, you need to do MCQs from 2018, 2019, 2020 until where you are right now, right? You will see that we have covered a lot of concepts, so you just need to apply. You just need to learn to apply those concepts in different clinical situations. Until next time.